today we will cover up the translocation topic, which is the movement of organic nutrient. Last week we talked about explain about the mechanism of transpiration, right? About the root pressure, transpiration pool, that. So it is talking about the transport of water from root to the leaf. So today's topic is about translocation, which is the transportation of organic nutrient from the leaf that produced from photosynthesis, like sucrose molecule, glucose molecule, and other organic nutrient that transport from leaf to whole part of the body. So this is a process we call as translocation. Okay, so normally this topic, okay, so as same as the last week, we know that transpiration process, the key organ that in the plant play the role is xylem, right? Xylem vessel. So for translocation, the key organ is also vascular tissue and is under phloem tissue. Phloem tissue will play a role in the key in transport of organic nutrient. So this is the phloem structure, right? So we know that in phloem tissue we have contain 4 types of cell, right? Component cell and sieve tube cell and parenchyma cell and also fiber. But for the cell that take place in the translocation is only companion cell and also sieve tube cell only. The parenchyma cell and also phloem fiber are not take part in the translocation. Only the sieve tube cell and the companion cell take part in the translocation only. So this one just recap. So everyone, you should know that you already learned in Sem 1, know that there are 4 types of phloem cell lah: sieve tube, component cell, fiber, and parenchyma. So only the sieve tube and component cell are take part in the translocation process only. Phloem fiber and phloem parenchyma are not take part as well. Okay, so this is the definition of the translocation. So as you see that translocation is the transport of the product from the photosynthesis, mainly is sucrose molecule or glucose molecule, that as from the sugar source to sugar sink by phloem tissue. So from here you need to know what is the meaning of sugar source and what is the meaning of sugar sink. Okay, so if you know that sugar source, you say that it is a source, right? Source meaning that it is an original place where the photosynthesis is synthesized. So normally the sugar source in the plant is leaf lah, because leaf is the places that the photosynthesis will take part, because they contains high concentration of chloroplast compared to other part of the plant. So sugar source is in the leaf, and then sugar sink is that normally is the place that your organic nutrient or your organic solute is utilized, it will be used or will be stored at there. So normally is whole part of the plant lah. So normally in root or in shoot lah, or in fruit. Fruit also is the sink of the plant. Okay, so normally in translocation we have in our syllabus we have 4 hypotheses that you need to learn. First one is about pressure flow hypothesis. Another one is electro-osmosis and cytoplasmic swimming, and lastly is about peristaltic wave. So the most famous one and the most common one that will be used is pressure flow, or another name we call as mass flow. So these two actually are similar, they are same, just different name only. Okay, so we already know that what is the sugar source and sugar sink. So normally this diagram is show you that how the sucrose that already produced in the leaf transported from the phloem in the leaf into the phloem tissue, sieve tube. meaning that it is the process that the sucrose molecule is already produced surrounding in the leaf any part lah. Because we know that in the leaf tissue we have palisade mesophyll and spongy mesophyll, right? And both of them containing chloroplast, especially palisade mesophyll. And then this part, and then when they contain chloroplast, they will undergo photosynthesis to produce the organic nutrient, right? So all the organic nutrient later will be transported into the phloem tissue, into the phloem tube, phloem sieve tube. So this diagram show you that how the sucrose molecule is be transported from the mesophyll cell into the leaf phloem tube. So actually, like the water molecule lah, water molecule in transpiration process, in water molecule you learn about the how the water molecule is absorbed from the root and transported from the root hair into the root xylem vessel, right? So similar for this is the process that you that the nutrient produced from in the leaf and then transported from the leaf into the leaf sieve tube element. So normally it will be all the organic nutrient firstly will be accumulated in the mesophyll cell first, because mesophyll cell is normally located nearly to the sieve tube, into the vascular tissue. So normally when the sucrose molecule is already accumulated in the mesophyll cell, it will be transported through the series of phloem cells. So normally if you see that there are many of mesophyll cell, and then bundle sheet cell, and also phloem parenchyma cell, and finally it will go enter the campanule cell too. Campanule cell. So we know that campanule cell, phloem parenchyma cell, and sieve tube element are the component of the phloem tissue, right? So normally when they are entering, when the sucrose molecule is already entering the parenchyma cell, some of the sucrose molecule will be entered through apoplast pathway. Some molecule, not all, some of them will enter the apoplast pathway, but the mostly pathway that will be used in the sucrose transport from the mesophyll cell into the phloem sieve tube is using symplast pathway lah. So they will finally enter the sieve tube element. So normally in this process, for those that transported into the apoplast pathway, they were using this core transport proton pump that require energy. Because if you see this diagram, you will see that the hydrogen ion inside the cell. So normally you will see here is inside the bundle sieve tube element. So the hydrogen ion inside the sieve tube element will pump the hydrogen ion out from the cell into the cell wall of the sieve tube element. So this one will be create the electrochemical gradient already. So this is one type of active transport, and then this is the core transport process. Also transport process. Why? Because the pumping of hydrogen ion into the cell wall will cause the electrochemical gradient. So that this now is highly energy, and then it will transport the sucrose molecule together down the concentration gradient. So that the sucrose molecule that accumulated at here will be together with the hydrogen ion pumped into the sieve tube element, the cytoplasm of sieve tube element. So this is one of the example of core transport process that you already learned in semester 1. Okay. So this is the explanation. So almost I already explained all. So normally if this is the very important, the sugar movement into the phloem require active transport. So normally compared to the water molecule normally is at passive transport. It is osmosis process, right? But in the translocation process, the sugar normally you need to use active transport process. Okay.